rest of this time talking about the plant experiment that I've been getting a lot of emails about, a lot of good questions about it too, about what's going on and some clarification questions just to make sure we're all on the same page, and then we turn in um, something on the first day of school that will give you the maximum number of points. So a lot of questions that I've been asking is, okay, well, what am I really supposed to do with this plant experiment? So you should have two seeds of the same plant, and you're supposed to grow both of them in identical conditions except for one different thing. That one thing that's different is your variable. Some individuals are deciding to keep in the exact same altitude, exact same soil, exact same type of plant, exact same amount of sunlight, but giving one of the plants water and giving the other plant soda. And you're supposed to grow them and see what the differences in plant height is after 2 weeks. So this 2 weeks that I've been talking about to a lot of individuals, it's not from the 2 weeks when you plant the seeds. It's once the plants break the topsoil, so you actually start seeing some of the plant, then you want to measure it for 2 weeks after that. That is what your time is. Your 2 weeks is once once you can start seeing the plant, and then measure how long it grows for 2 weeks. You don't need to keep growing it for the entire summer. You just need to do it for 2 weeks. If you did not pick up a seed from me during the last week of school, I was passing out sunflower seeds. You can go to Lowe's or Home Depot or any other type of place to get a fast growing plant. That's what you want, a fast growing plant, because some plants, they take weeks or even months to break the topsoil. So you want to get these fast growing plants so they break the topsoil. Quickly so you can finish the assignment before the first week of school starts. Some other questions I've been getting is, okay, so I got my plant, I did the experiment, I changed only one thing, and that is my variable that I'm talking about, but in the results, it's saying what are these two different independent and dependent variables, okay? Well, the independent variable is what doesn't depend upon anything else, and that's time. And this is different from the variable I was talking about because that's what you changed earlier. Everything that you keep the same, like the type of soil or the amount of sunlight, that's called your constant, and then the variable is the one thing you change, whether you gave the plant water or Coke, going back to my earlier example. And then now when we're in the results part and we're graphing it out, it talks about an independent and dependent variable. Your independent variable is time, which is always on your x-axis. Always on your x-axis because time doesn't depend upon anything else. And your dependent variable is the plant height, because depending upon how long you've been growing your plant, that's what's going to determine how big your plant height is. So when you're making the graph of the two different plants, you want to say, oh, okay, after 5 days, plant 1 with the water grew this many centimeters, or this many meters, or this many miles. It is up to you, the units. For plant 2, then you want to have another graph saying, okay, so this is my x-axis, these are the number days, and this is how much it grew after 5 days. And again, you want to keep it the same units as plant 1, but it could be any units you want. And then now going to the lab report rubric, I'm looking on my rubric over here on the side. Is talking about mini poster rubric. Well, what does a mini poster look like? The size that I'm looking for is not something huge. So this is a manila folder, and when you open it up, it's one of these little folders with a little tab in it, and I got one of these. Open it up, not too big, and then here I have 2. And then so with the 2, what you want to do, you should be able to open them up. We'll keep the Office Max plug on the inside. You want to open them up, and then you want to open them up, and then you can just tape them together on the inside. And this is about the size that I'm looking for. It's nothing too big, nothing too small either, and this should be enough to show you all your different parts. So once you have this together, 2 of these folders, and again, it doesn't have to be these folders. You can just get one of those big poster boards and just cut it in half, which would be about the same size. Once you have this, you want to have, like up here, you can have whatever title you want, and then looking at the components of the different rubric that I'm looking at over here, you can go like question, you want to like put a title, and then you want to write down what your question is. Again, this could all be handwritten, or it could be typed out and cut and paste, whatever you think will look best. And then you talk about your variables. Sorry about that. And then you want to talk about your hypothesis, the materials, procedures, all that sort of stuff, just post it up on here. Whatever you want should be able to fit on here. That way you don't have to make it too big, or and that way it won't be too small either. This is about the size that I'm looking for. Some other questions had to do graphs, no conclusion. Error analysis. So what exactly does error analysis mean? Well, here you can be talking about what do you think you could have done by accident or that was out of your control that could have altered the results in any way. And again, it's just a discussion. What you think you could have, what you think could have gone wrong, or what you think might have changed your results. And another procedures, materials. Oh, pictures and diagrams. I've been getting a lot of questions about that. That is up to you. I'm not looking for your procedures to have like a picture or a diagram for every single step of the procedures that you make. As long as you just have one or even two diagrams or pictures that show the overall process, that's all I'm really looking for. It doesn't have to be anything too complicated. Again, this is not going to be the most complicated lab experiment we're going to do this year. This is just something simple, just to get our feet wet, just to get started in this whole scientific method and biological process. And yeah, I think that's about it. And if you still have any more questions, please feel free to email me. I've been getting a ton of emails. Usually I've been very good about responding to emails. I've been traveling a little bit, so during those times I usually get back within 48 hours. But if you email me and I'm here working, which I'm working a lot on just getting ready for school and doing a bunch of other stuff, I should be able to respond to your email within the same day. I'm usually pretty good about that. So don't worry about reaching out to me. I'm not going to think like, oh, you don't get it, why are you asking me these questions? No, I love these questions. I've been getting a lot of good questions, and I want to keep hearing these good questions just to make sure we're all on the same page.